The next important technique is DNA fingerprinting. In DNA fingerprinting, you can see what is the steps involved in DNA fingerprinting. So first we should know what is DNA fingerprinting technique. So this DNA fingerprinting technique was developed by Alex Jeffrey in the year 1985. So each of us have the same chemical structure of DNA, but there are millions of differences in the DNA sequence of base pairs. This makes the uniqueness among us so that each of us accept identical except the identical twins is different from each other genetically. So the DNA of a person and fingerprints are unique. So we will see how this DNA fingerprinting technique is done. So here the blood sample is collected and the DNA is isolated from the blood sample. So the blood cells, RBC or whatever the cells present in the blood, from that the DNA is isolated, and this isolated DNA, with the help of the restriction enzymes, it is digested and cut into small fragments. So fragments of DNA you can see here. These fragments are separated by electrophoresis, that is agarose gel electrophoresis method. So these fragments, when it is sent to the agarose gel electrophoresis, it will be separated like this. Then transfer of DNA fragments to the membrane, that is using southern blot technique. You learn this electrophoresis and southern blot technique in detail in botany. So this is the electrophoresis method where the DNA fragments will be separated. After that, this will be copied, or you can say this will be made to attach to the membrane by using the southern blot technique. After that, this DNA fragments will be hybridized with radioactive probe. So the single strands will be made to double strands now. After that, excess radioactive probe is washed. So whichever is additional, whichever are not binding to this, that will be washed out. Then developing the X-ray film to detect the radioactive probe. So to whichever that probe is bounded, they all will show radioactivity. So developing the X-ray film to detect the radioactive probe. Then DNA pattern is compared with the patterns of known subjects. So whomever is found to be suspect. So with that, the DNA whichever we have obtained, with that they will identify whether it is equivalent, that is it is matching to any of this. So they will be able to identify the particular suspects also. So this is how the DNA fingerprinting is done. Applications of DNA fingerprinting. It is used in forensic analysis, that is to detect crime, and for settling parental disputes, that is to find out who is the father or mother of a particular child, and also for immigration purpose, that is when you go out of the country. Then pedigree analysis, that is inherited pattern of genes, to know the inherited, how the genes are inherited, from whom the genes are accepted or inherited, to detect inherited diseases, from where the diseases have been inherited. To know that also, this fingerprinting, DNA fingerprinting helps us. Next one is conservation of wildlife. So for protection of endangered species. So the particular species which is about to be extinct, so that particular gene or DNA can be conserved with the help of this DNA fingerprinting, that can be known or can conserve that. Next one, anthropological studies, determining the origin and migration of human population and genetic diversities. So how the human race are widespread, how from where man originated. So all to know about the human population also, this DNA fingerprinting helps us. Now let's see a video about DNA fingerprinting technique and its applications. Every person has two unique fingerprints, the pattern on their fingertips and the pattern of their DNA. Fingerprints can be altered by a surgery, but a person's DNA fingerprint is the same in each of their cells. It cannot be altered by any known treatment. Let's see the complete guide of DNA fingerprint technique. DNA fingerprinting is a technique to identify people using samples of their DNA. The DNA fingerprint consists of the pattern of DNA fragments which are obtained by restriction endonuclease enzyme. DNA fragments within the genome whose number and arrangement are unique for each person. The technique of DNA fingerprint was used for the first time by Dr. Alex Jeffrey in 1984. Important for DNA fingerprint are short nucleotide repeats which vary in number from person to person and are identical. They are called variable number of tandem repeats or VNTRS. Now let's see the steps of DNA fingerprinting. The first step, a sample of DNA is taken from blood, hair follicle, saliva, semen or any body tissue cells. In many situations, only very small amount of DNA is available for DNA fingerprinting. Because of this, PCR or polymer chain reaction is applied. After the DNA is isolated and more copies of DNA have been made, the DNA is treated with restriction enzyme. This will produce different fragment size known as RFLPs, that is restriction fragment length polymorphism. DNA fragments are then sorted according in size using gel electrophoresis process. During this process, DNA fragments are injected into well in agarose gel and an electric current is applied along the gel. DNA is negatively charged, so it is attracted towards the positive end of the gel. The shorter DNA fragments run faster than the larger one. Now the DNA fragments are transferred to a nitrocellulose paper by pressing the sheet on the gel and soaking them overnight. Now a radioactive material is added which combined with the DNA fragment to produce fluorescent image. Lastly, the nitrocellulose paper is placed against the piece of X-ray film. The X-ray film can record the pattern of radioactivity on the paper. Next is applications of DNA fingerprinting. So to find out the disputes, that's paternity testing. It is also useful as identification tool in forensic application. It helps in diagnosis of inherited disorder. It is used to determine population, to know genetic diversity in evolutionary biology. So these are the applications of DNA fingerprinting. So we have come to the end of lesson 5, molecular genetics, and this last 4 topics, translation, regulation of gene expression and human genome project, DNA fingerprinting. These 4 are very, very important questions. So as I told you before in the introduction itself. So you will have a test in any one of these questions in the evening. So either in translation or regulation of human genome project or DNA fingerprinting. So go through these slides carefully and you can start reading. You will have a test in any one of these